Hello, good day everyone. My name is Angelica de la Cruz, and for today's video, we will tackle about the ethical issues on gene editing in human embryo. So, despite the fact that the potential benefits of clinical germline genome editing or CGA over currently available technologies are limited, CGE in the clinic has been proposed and debated. So first, let's define what is genome editing. It is a powerful new tool for making precise addition, deletions, and alterations to the genome, an organism's complete set of genetic material. So these technologies allow genetic material to be added, removed, or altered at a particular location in the genome. So several approaches to genome editing have been developed. One that the scientists use that genome editing is to investigate different diseases that affects the humans. So they edit genomes of animals like mice, zebrafish, because animals have many of the same genes as of humans. Now, let's talk about what is CRISPR case 9, which is a short for clustered regularly interspaced short palindromic repeats, and CRISPR associated protein 9, a system that allows for precise, efficient, relatively cheap and fast modification of DNA in various organisms and types of cells. Genome editing has been found to have applications in many research areas, including, among others, gene function studies, gene therapy studies, drug development, and production of modified plants. As with other medical advances, each applications come with its own set of benefits, risks, regulatory questions, and ethical issues and societal implications. Important questions raised with respect to genome editing include how to balance potential benefits against the risk of unintended harms, how to govern the use of these technologies, how to incorporate social values into salient clinical policy considerations. So in 2015, the first experiment with CRISPR Cas9 on human embryo has reported. The authors demonstrated that genome editing system performed targeted cleavage in the B globin gene, which when muted causes B thalassemia. Following this publication, discussion on prospective clinical applications on germline genome editing or CGA in humans and related ethical issues was ignited. So most of the changes introduced with genome editing are limited to somatic cells, which are cells other than egg and sperm cells in germline cells. So, the use of embryo in research raises a number of ethical aspects. One commonly discussed ethical issue is that related to the destruction of human embryos. So, furthermore, we may distinguish viable and non-viable embryos. The former term means that embryos are considered to be able to develop normally and result in a live birth when implanted into woman's uterus. And non-viable embryos, example tripronuclear embryos, are considered unable to develop normally, and the result is a live birth if implanted into a woman's uterus. So, there are three main positions in discussion on the moral status or value of the human embryo. A, we have human embryos have the same moral status as any other born human. B, human embryos have some moral status or value that not the same as a born human. There are variations within this view. For example, some say that moral status or value of embryos increase during their development. And letter C, human embryos have no moral status or their moral status value is the same as any other type of human cells. So, if we assume the position that human embryos have the same moral status as persons, CGA experiments are considered unacceptable. So, research on embryos is prohibited in some countries like Austria, Germany, Italy, Poland. However, the formulation of this legislation differ. In the studies on the CGA, both viable and non-viable embryos have been used. In one study by MA et al. In 2017, embryos were created specifically for the purpose of research. And notably, the usual number of embryos used in such procedure can be high. And for instance, in the study conducted by MA et al. 2017, over 100 embryos or human embryos were created and destroyed. The continuation of such experiment would certainly multiply these numbers. CGA research using embryos raises questions about the moral status of the human embryo, the involvement of the egg donation, which entails serious risk to women. So that is all for today. I hope you learned something about the ethical issues on gene editing on human embryo.